Thank you for being here. My name is Angeline Lacson, and today I'm going to share with you the results of my thesis project, which is entitled A Multimetric Approach to Evaluate Offshore Mussel Aquaculture Effects on Receiving Benthic Communities, Are Natural Factors Masking the Induced Impacts. My thesis project was a response to the growing need to understand the effect of aquaculture on the marine environment and the need to minimize these effects. Offshore aquaculture has been proposed as a means to achieve this need, but there remains the question of its sustainability. In the case of offshore mussel aquaculture, the impacts are associated with byproducts organically enriching the underlying seafloor. How does this affect the macrobenthic communities? Would this alter their habitats in such a way that would induce a scenario of anoxia, for example? Would this then change the structure and composition of the macrobenthos? And could this induce shifts in the functional ecology of the area? The Marine Stewardship Council is concerned with these effects, and one of the principles behind their sustainable fisheries certification states that the fishery should allow for the maintenance of the structure, productivity, function, and diversity of the ecosystem. In line with this, my project focused on an aquaculture production area, or APA, in southern Portugal, in the Algarve area. My primary aim was to detect with a battery of statistical tests if the APA induced changes in the species composition and functional ecology of the underlying communities. And because it was a multimetric analysis, I made use of several hypotheses. I'm not going to read each one of them, but they have to do with differences in the concentrations of organic matter and metals, the composition of macrobenthic communities, alpha and beta diversity patterns, and the functional structure and organization of the local macrobenthic communities. One of my hypotheses also highlighted the potential of natural factors such as depth and hydrodynamics in shaping the local macrobenthic communities. So this is supposed to be a video showing the harvesting of the farm, which was a semi-submerged long-line mussel farm of Mediterranean mussels. And each of the long lines were around 400 meters long with 150 headlines each, and each of the headlines were approximately 12 meters high. And the farm would produce around 4,000 tons of mussels, and the total farm area was around 15 square kilometers. So the gray box shows the area of the farm, which was, located close to Armona Island in the south of Portugal. And in this area, you have currents coming from the southwest and the southeast, which is why at the time of sampling, the persons responsible for conducting the survey decided to put sites all around the farm instead of just one area. So to comprehensively evaluate the effects of the APA, I made use of a BASI, or before-after control impact approach. I also divided the sites into shallow and deep areas to account for differences in hydrography, because in Portugal, wave effects extend to areas that are around 30 meters deep. So below the blue line, you have areas that are around 40 to 70 meters deep. So it's understood that the macrobenthic communities would be differentially affected by the hydrographic conditions. So there were 6 control sites outside of the farm and 6 impact sites inside of the farm. And there were also 4 periods of sampling. 2 of them were before the mussels were established in the farm in the year 2010, and 2 of them after the establishment of the farm in 2014. From each sampling period, Van Veen grab was used to collect sediment, and from these sediment samples, the organic matter, the sediment fine fraction, and the metal concentrations were quantified. And the macrobenthic taxa from these samples were also identified to the lowest possible taxonomic level. My multimetric analysis combined classical and innovative approaches so that I could fully understand the functional ecology of the area. The classical approach included multivariate statistics such as MDS, Permanova, and Simper, and alpha diversity metrics to obtain the number of individuals, species richness, Shannon-Wiener diversity, and evenness in each station. I complemented these tests with beta diversity, which is defined as the difference in diversity between two different sites. So I made a schematic to explain what the concept was to you in case it's a little bit new to you. Every single site has the same alpha diversity because there are 3 species and 3 individuals per site, whereas the beta diversity for sites B and C is higher than B and A because you have more species between sites B and C, for example. And the beta diversity scores, which I obtained through Sorensen dissimilarity, can be partitioned into turnover and nestedness components. Turnover is basically defined as natural species replacement, and it's a phenomenon that's observed in undisturbed communities, where communities just gradually change over time or space. Whereas nestedness is defined as a phenomenon of species loss, where over time you have species that disappear from the community. And this is normally found in disturbed communities. I also made use of a functional diversity approach, and to do this, I conducted a bibliographic survey of biological traits, or a biological traits analysis. I made use of these biological traits, and basically I had to go online to sites like worms to obtain information about the species that were found. So for my traits matrix, I made use of a fuzzy coding technique where each modality was given a score of 0 to 3, with 3 being a full affinity towards the expression of that modality and 0 being no affinity. So for example, if you look at the first row, you see Spicula solida, which is a bivalve, and it's exclusively a suspension feeder, so it's given a score of 3. And I did this for every single biological trait for each of their modalities. And then I made use of this data for an RLQ and fourth corner analysis, which looked for non-random relationships in the data so that specific environmental parameters could be related to specific biological traits. So let's proceed to the results. A total of 17,868 organisms belonging to 601 taxa were identified, and these were predominantly polychaetes, bivalves, and amphipods. All of the sediment descriptors actually increased with depth independently from the farm. And this makes sense because deep areas tend to have finer sediments because of the reduced effects of wave action. And organic matter and metals tend to have an affinity with fine sediments. It also followed that the communities were partitioned by depth instead of their positions relative to the farm. In terms of alpha diversity, the encompassing effects of depth were again seen. So as you can see here, this is the shallow control area, shallow impact depth, deep control, and deep impact area. And they tend to have relatively the same temporal trends between the impact and the shallow areas, apart from, for example, the deep impact area where you actually see a higher species richness after the establishment of the farm. And the same was seen for species diversity and evenness, where you have generally a higher species diversity in the shallow areas, generally a higher generally a higher evenness score for the deep areas. And tests that have previously seen scenarios like this say that it's possible that for example, shells that fall off of the farm can actually contribute to the complexity of the area, opening up new niches. Although in my case, these results were not statistically significant. As for beta diversity, the scores were generally similar across for each area temporally. However, a dominant profile of turnover was seen for all of the areas, which is good because it means that the sites were largely undisturbed. Although there was a slightly higher expression of nestedness in the impact areas, this was already detected even prior to the establishment of the farm. So this could have been a result of like historical farming activities that took place in the APA instead of the mussel farming activity itself, considering that this component was gradually decreased over time. So the functional trait analysis again showed that the communities were divided according to depth rather than their position relative to the farm. This first axis explained 98.3% of the variance. And if you have a look at these specific traits, for example, body size and larval type, you can see that in the shallow area, you have the same temporal expression of traits. And in the deep area, you have the same again. And I saw the same type of expression for all of the other functional traits. If we have a look at the RLQ projection of traits and species, we again see typical relationships between biological traits and environmental conditions. Keep in mind that the right side of the graph is related to the shallow areas, which had coarser sand and lower organic matter, whereas the left side of the graph was representative of the deep areas, which had higher organic matter and metal concentrations. So in the shallow areas, there were more suspension feeders, such as Spicula solida. There were also more epibenthic organisms, such as this hermit crab, Diogenes pugillator. And there were also more short-lived, small brooding organisms, represented by amphipods of the genera Eurotho and Photis. In the deep areas, on the other hand, there were more surface deposit feeders that were also burrowers with a planktotrophic type of larvae. And these were more represented by polychaetes of the species Heteromastus filiformis, Lagus careni, and Glycera unicornis. And these polychaetes are actually, they have the tendency to be opportunistic or tolerant, although we have to remember that the functional traits in the impact sites were the same as that of the control sites and even that of the pre-mussel scenario. So it's not exactly conclusive that the byproducts actually went down the slope and accumulated in the deeper areas because the species were already there even before the mussel farm was established. So again, depth-related conditions were shown to establish the functional structure of the area. Ultimately, the results suggested that the APA did not adversely impact the seafloor. Depth was seen to supersede the effects of the APA, as it was shown to shape the environment, the community composition, and the functional diversity in the area. The fact that turnover was the dominant component of beta diversity also showed that the ecology was not influenced in a way that would induce species loss, but rather species replacement over time. And these results highlighted the potential of an offshore profile because hydrodynamics in offshore sites are maybe strong enough to disperse the byproducts of the farm instead of causing a scenario in which a mass settlement of the byproducts would occur. It remains essential, however, to continuously investigate the ecological structures under offshore farms so that we can fully comprehend the long-term impact of offshore farming. And this, combined with the expansion of reliable trait data and the integration of other disciplines, would perhaps improve the way that offshore farms can be managed and developed in the future. Thank you for listening. I'd like to thank my promoter and my supervisor and the Portuguese Institute of the Sea and Atmosphere. And yeah, that's all. Thank you.